Welcome everyone to today's session. We're very happy to have you join us. My name is Marie Latulippe and I manage some of the nutrition programs at our organization. And today we have two excellent speakers and we hope to provide you with some tools to enhance your practice and perhaps respond to your patient and client questions about sweetness in the diet. So first, for those of you who might not be familiar with our organization, we are a nonprofit scientific organization that advances the food safety and nutrition science for the benefit of the health of the public. And the way we do that is we bring together scientists from industry, academia, and government, and together they determine what the research questions of common concern might be and what are the approaches to those questions. And we have about 12 nutrition-specific committees at our organization. One of those is the Low-Calorie Sweeteners Committee that brings you this webinar today. And you can see their mission on the slide here, but they're very interested in providing science-based information to practitioners. So that is why we've developed this webinar series, and this is about the third in that series. So this webinar is approved for one continuing education credit by the Commission on Dietetic Registration. And after the webinar, you will receive a really brief survey. Just complete that survey and we will get the certificate to you. And as usual for all these webinars that we're all on lately, feel free to type your question into the chat box at any time over the course of the webinar. And what we plan to do is get to those questions at the end. So with that, I'm very pleased to introduce our first speaker, and that is Dr. Beverly Tepper. Dr. Tepper holds a PhD from the Tufts School of Nutrition and Science Policy, and she's been a professor of sensory science and nutrition in the Department of Food Science at Rutgers since 1989. Her research examines the intersection between taste, psychology, and nutrition. And she's best known for her work on the genetics of taste and its role in food selection and body weight. She was also recently elected a fellow to the Institute of Food Technologists, and she currently serves as the scientific editor of the Sensory and Consumer Sciences section of the Journal of Food Science. So with that, we are very happy to turn the platform over to you, Dr. Tepper.
Thank you so much. Finding my webcam. Give me a moment to get myself organized. And perfect. Okay, good afternoon, everybody. Thank you, Marie. And thank you, Ilse, for inviting me to present this afternoon. I am so delighted that you're having such an enthusiastic group of attendees today. It indicates that this is really an area that a lot of nutrition folks have a great interest in. So today I'm going to be talking about individual differences in sweet taste and their implication for nutrition practitioners. So my overview will be, I'm going to first talk very briefly about non-nutritive sweeteners. I'm going to talk a little bit about how sweet and bitter tastes are perceived, because both of those are very relevant for non-nutritive sweeteners. I'll talk about the role of individual differences in perception and liking of sweet, focusing on genetic differences in taste, and then talk a little bit about the concept of a sweet liker and disliker. And then lastly, I'd like to share with you some results of a study that we did a number of years ago looking at sweet taste in gestational diabetes. So these are the major sweeteners approved for use in food products in the US. Of course, we have our representative of carbohydrate sweetener, sucrose, and as well the various sweeteners that are currently approved. These sweeteners can be between 200 and 300 times as sweet as sucrose, with the exception of sucralose, which is 600 times as sweet as sucrose. We know that these sweeteners are, most of these sweeteners are artificial in nature, except for stevia, which is a natural sweetener, the most recent one added to the group of approved sweeteners. And this comes from the stevia plant. So what are the challenges for sugar substitutes? We call sucrose, fructose, and other carbohydrate sweeteners bulk sweeteners, and they provide what food scientists call functionality. They help to control various aspects of texture as well as other aspects of a food product. And this can involve controlling thickness and viscosity, such as in sauces and syrups that you want to flow freely, the moistness of the crumb of cakes, the creaminess of ice cream and confections, as well as the crispy, crunchy outside chocolate coatings of ice cream products and confections. So when we remove the sugar from a food product, we also reduce all of this functionality. So when you make those substitutions with non-nutritive sweeteners, you've lost these functionalities. So many other ingredients need to be used to replace these characteristics. The other aspect is that non-nutritive sweeteners lack what we call a clean sweet taste. So what I mean by a clean sweet taste is that sucrose, for example, shows a specific temporal pattern in how you perceive it on the tongue. And I'll show you an example of this in just a moment. The other aspect is that non-nutritive sweeteners, generally speaking, impart bitterness, which can be a challenge for consumer liking of non-nutritive sweeteners. So this is a sweet taste profile of 3 different sweeteners. And what you're looking at here is the increase in intensity over time with sucrose, saccharin, and this is an experimental non-nutritive sweetener. So we can see for sucrose, the sweetness increases very rapidly. It reaches its peak, and then it decreases very rapidly. A nice, clean, sweet taste. Saccharin, we see it increases very rapidly, and there's a little bit of persistence that occurs with this sweetener. And if we look at this third sweetener, NHDC, we see there's a delay in the increase in sweetness. It reaches its peak, and then it decreases very, very slowly over time. So this creates a persistence of sweetness in the mouth that can be quite noticeable over time. This is the TAS1R sweet receptor. So we can see this is a very complex structure that would be sitting in a cell membrane. This would be the inside of the cell. This would be the outside of the cell. And what we can see is that the various sweeteners can bind to this receptor, although they bind to different locations of this receptor. As far as we know, humans have one type of sweet receptor. But we also know that non-nutritive sweeteners interact with both the sweet receptor and bitter receptors. So what you see here, this is the same sweet receptor that I showed you in the previous slide, and next to it is a bitter receptor. And what we can see is that this saccharin molecule is binding to the sweet receptor. It is activating that sweet receptor, but it's also activating this bitter receptor. So saccharin is well known to have a very intense sweet taste, as well as a very intense bitter taste to some people. And we'll come back to that concept in a moment. This is a molecule of cyclamate. Cyclamate is not currently on the list of approved non-nutritive sweeteners. It was removed a number of years ago, but it tells the story here. So cyclamate activates the sweet receptor, but it blocks the bitter receptor. So non-nutritive sweeteners are often used in blends to take advantage of the fact that two different kinds of non-nutritive sweeteners will synergize with each other. You get more bang for the sweetness buck by using two types of sweeteners together. And then you have the advantage of the cyclamate blocking some of the bitterness of the saccharin. It's well known, for example, that aspartame and acesulfame-K blends are commonly used in soft drinks. So if you look at the list of ingredients on many soft drinks, you will see that both of these non-nutritive sweeteners are used. Oh, I wanted to make one comment here. Humans have approximately 25 types of bitter receptors. Why would that be the case? Well, there are many compounds in nature that are bitter and also toxic. And this is very relevant for various types of plants. So 10,000 years ago, when we foraged for our food, it was very, very important for humans to be able to perceive bitterness wherever it may be in the environment, and to have receptors that are able to respond to any types of these compounds. Some bitter receptors only respond to a very short list of related compounds. Other bitter receptors respond to a wide array of bitter compounds. And by having this whole net of different bitter receptors, we sort of have an insurance policy that we will be able to perceive any kind of bitter compound that we might come in contact with. Here are a list of intense sweeteners that activate different types of bitter receptors. So what I've highlighted here are some of these examples. So acesulfame-K activates the TAS2R9, 43, and 31 receptor. All of these receptors have numbers. Cyclamate activates these 4 receptors. Likewise, saccharin, these 3 receptors, and stevia, these 2 receptors. And one thing you'll notice that this TAS2R43 here, here, and here is activated by 3 different kinds of non-nutritive sweeteners. So in addition to the fact that we have many different kinds of bitter receptors, we also know that we have genetic variation in these bitter receptors, so that much of the individual differences that we see between people and how they perceive bitter and these sweeteners has to do with variation in the genes that they have for these receptors. So, for example, there have been studies looking at TAS2R3, 4, and 5, and how individuals who express different variation in those receptors perceive differences in the bitterness of espresso coffee. Similar work has been done on receptor TAS2R19 for quinine in tonic water and grapefruit juice. We've already looked at non-nutritive sweeteners and the fact that they interact with specific receptors There's genetic variation there too. And the last one I want to mention is TAS2R38. This is the receptor that I study. And this one is a little bit of a special receptor because it controls the ability to perceive the bitter taste of 6-M-propylthiouracil, or PROP, which is an index of general oral sensations. So I'd like to talk a little bit about this in the next slide. So what is PROP? PROP is a bitter marker for genetic variation among people and how they perceive bitter taste. So PROP is similar to naturally occurring glucosinolates that are in cruciferous vegetables. So we know many people dislike cruciferous vegetables because they're bitter tasting, such as broccoli, Brussels sprouts, cabbage, and so on. And it's the glucosinolates that play a role in this. So there is genetic variation in the gene that controls PROP tasting. And we can divide people basically into 3 very discrete groups. 30% of people are non-tasters of PROP. They're taste blind to this compound. They taste little or no bitterness from it. About 45% are medium tasters. They find PROP to be moderately bitter. And about a quarter of the population are super tasters who perceive extreme bitterness from this compound. It's quite easy to measure a person's PROP taster status using a laboratory solution, or here we use filter paper discs impregnated with PROP. I can talk to you more about those screening techniques after we're done with the formal presentations, if you like. But why is this important? So we've come to recognize that PROP tasters and non-tasters really live in very different taste worlds. PROP tasters are more sensitive to bitterness and sweetness, which is the focus of what we're talking about here today, although they're also more sensitive to pungency, like the hotness of chili pepper, as well as astringency and fat. Non-tasters are less sensitive to these qualities. So what this means is that, generally speaking, non-tasters tend to show increased preferences for foods with these qualities. They tend to like things more sweet and to accept things with higher bitter quality, whereas tasters, and especially super tasters, show the opposite responses. They show decreased preferences for foods with these qualities. So what does this have to do with non-nutritive sweeteners? There have been some studies over the years looking at the role of PROP taster status and perception and acceptance of non-nutritive sweeteners. We have some historic data from many, many years ago looking at saccharin, showing that super tasters perceive more sweetness as well as more bitterness from saccharin. We've already talked about the sweet and bitter aspects of saccharin. And generally speaking, for people who are super tasters, saccharin is a deal breaker because the bitter aspects tend to be quite strong for them. We did a study a number of years ago looking at citrus beverages where we use blends of different non-nutritive sweeteners, and found that super tasters perceive more persistence of sweetness, persistence of bitterness, as well as aftertaste from these sweeteners. Unfortunately, in this study, liking wasn't affected, which would be an important outcome, but the degree of liking of this beverage was rather neutral for our subjects. So none of the subjects thought that our formulations were that great. So we need to go back and look at this again. Not all studies come to the conclusion that PROP plays a role in perception and acceptance of non-nutritive sweeteners. But more research is needed on this topic because, as you can see, there's only a handful of studies that have really looked at this issue. Well, what if we combine PROP status with another measure of individual differences? Will that give us a better read on people's acceptance of non-nutritive sweeteners? So what I want to introduce now is the concept of sweet liking and disliking, and Robin is going to expand on this topic in her presentation. So what is a sweet liker and disliker? Sweet liker is a person who shows increased liking with increasing concentration of sweet stimulus, and a sweet disliker shows decreased liking with increasing concentration of a sweetener. And studies have shown that this is useful for characterizing individuals' reactions to sweet stimuli. And this is an example from a study from Yeomans. So we have our sweet likers here. We see they're showing higher and higher liking responses as the concentration of sucrose is increasing. And here we have our dislikers showing the opposite response, where they are showing lower and lower liking as sweetness is increasing. And what I'm indicating here, so let me just say this is in millimolar concentrations, which is not useful to this audience. But if you translate that into percentage sucrose, this concentration is a 26% sucrose concentration. This is like mind-numbingly sweet. Most food products are in this range, maybe 8 to 10, at most maybe 12% sucrose. So you can see that the sweet likers are expressing a very strong liking for extremely sweet stimuli. So what if we combined the sweet liker disliker classification with PROP tasting? And this is also data from the Yeomans study. And what we can see here is we have non-tasters, medium tasters, and super tasters. And we can see that the non-tasters are the sweet likers. They're increasing over sucrose concentration. We have medium tasters who are neutral across concentrations. And then we have super tasters who are showing the dislike, increasing dislike at increasing concentrations. So this shows us that using both classifications may be more predictive, may give us a better read on food acceptance than either of those measures alone. So let me move on to my last topic. We had an opportunity a number of years ago to do a study looking at changes in sweet taste in gestational diabetes. This audience well knows that type 2 diabetes and gestational diabetes have the same underlying metabolic dysfunction. It's insulin resistance that leads to increased plasma glucose levels. In the case of gestational diabetes, the diabetes is showing itself during pregnancy. So there's data showing impaired perception of sugar in type 2 diabetics. We did a study a number of years ago looking at intake of sweet foods in type 2 diabetics, and they show higher intake of sweet foods and also higher intake of non-nutritive sweeteners. So they may be an open audience for the use of non-nutritive sweeteners. From the pregnancy literature, we know that increased cravings for sweets occur in normal pregnancy, and they tend to peak in mid-pregnancy and then decline towards delivery. So we asked the question, do women with gestational diabetes have higher preferences and cravings for sweet foods than women without GDM that could undermine their diet adherence? So we did the following study where we followed women across their pregnancy. We did 3-day diet records at early, mid, and late pregnancy, as well as early postpartum. The subjects in the study were women who eventually were diagnosed with gestational diabetes, pregnant women with normal glucose tolerance, and then non-pregnant controls. And we looked at all sweet foods and beverages, fruit and fruit juice, and then other sweet foods and beverages. And what we found was the normal glucose tolerant women showed a peak in intake of sweet foods in mid-pregnancy, as the literature would suggest. We found no increase in the women with gestational diabetes. But it's important to note that these women had mild gestational diabetes and they were controlling their disease by diet. So they were quite compliant and also had good glucose control over the course of their pregnancy. However, let's turn our attention to cravings. We see a very different story emerge. We can see that in late pregnancy, the women with gestational diabetes showed a very high peak in their sweet cravings. Why did this come out very late in pregnancy? We don't know the reason. Is it biological? We don't know. Is it because they've been maintaining their diet all through their pregnancy and they just, hit a wall towards late pregnancy? We don't know. But an important aspect of this is that only 40% of the women with GDM had frequent cravings, but they accounted for most of the sweet cravings among women in this study. So what that is telling us is there's a small number of women with GDM who experience very acute sweet cravings late in pregnancy, and perhaps it might be useful to track sweet cravings across pregnancy as a way to help monitor and understand these women. So let me summarize what I talked about today. Understanding bitter taste is important for assessing and understanding acceptance of non-nutritive sweeteners by consumers. There are large individual differences in perception and acceptance of sweet taste. Today, we looked at genetic variation as it relates to pro-taster status. We introduced the idea of the sweet liker-disliker phenotype and how those two factors may lead to more insights. And we looked at sweet cravings and intake in women with GDM and their implications for disease management. So to pull this all together, I would suggest that these factors may be useful to practitioners to better match diet advice to individual clients' needs and preferences. And thank you so much for your attention.
Great, excellent. Thank you, Dr. Tepper. I think we'll go straight to the next speaker and take the questions at the end. So I am very pleased to introduce Dr. Robin Tucker. Dr. Tucker is an assistant professor in the Department of Food Science and Human Nutrition at Michigan State University. She studies factors that influence food choice, including taste. And some of her recent work has explored how sweet taste perception changes under conditions of insufficient sleep, which maybe some of us are familiar with. So with that, I will turn it over to you, Dr. Tucker.
Thank you so much, Marie and Ilse, for the opportunity to talk to you a little bit today about some of the struggles we face when trying to kind of craft some health messages around sweet taste. So today I'll go over a couple of topics. The first, we'll talk about the disconnect between the public messages surrounding sweet taste and the public health messages about sweet taste. And as public health practitioners and dietitians, we're fighting this battle that the public thinks that if it tastes good, it simply can't be good for you. I don't know if you're familiar with the website Quora, but it's a website where you can go and post questions, and then supposedly experts will respond to answer your question. And you can see here we have questions like, why do unhealthy foods taste better than healthy foods? Why does healthy food taste bad but unhealthy food tastes good? And there are over 100 answers. So clearly this is a matter of debate and interest among the public and for us too as nutrition professionals. And then we'll also sort of move into why is it challenging to make these predictions when we know something about sweet taste, liking, and preference? Why doesn't that always translate into solid predictions about dietary intake or quality as well as weight status? And as Dr. Tepper has alluded to, these individual differences can make our conclusions a lot more challenging because we've got people kind of going in different directions. And that's where we'll talk a little bit more about sweet liking phenotypes. And then I'll wrap things up with just a brief discussion about non-nutritive sweeteners. Again, just to kind of remind you or to build upon what Dr. Tepper mentioned, what's the function of sweet taste? Why do we have this sensory ability? And back in the day when food was scarce, we think that sweet taste was an important indicator of energy, particularly coming from carbohydrate, but energy in the environment. And because it was pleasant or pleasurable, that gave us sort of the go-ahead, the signal that this is probably safe for us and nutritious and good for us to eat. And so we would be able to meet our energy requirements and go on to live, happy, fulfilled lives. Okay, so sweet taste signal that there is energy available in the environment and it's probably not going to kill us. So it's an important sensory tool. Now, when we think about sweetness in terms of the mind of the public, they are very focused on the negative aspects of sweetness. And I want to just remind us all that this is more nuanced than that. But you see headlines about what to know about sugar in fruit, 4 vegetables you wouldn't think contain sugar. Is there any good sweet stuff that's not bad for you? 11 sweet and delicious things that aren't that bad for you. There's promise, right? And is it ever okay to eat sugar? Now, these are designed for clicks, right? So I read these sensational headlines and I'm gonna click on this and see. And in some cases, the articles do distinguish between naturally occurring sugars like those in fruits and vegetables versus added sugars, but in some cases they don't. And I think the important thing to remember here is that 60% of Americans don't read past the headline in many cases. So if I'm getting bombarded with messages about sugar in fruit and sugar in vegetables, I might start to think that those are not good choices for me, and I may adjust my intake accordingly. And that is not great for most Americans. The public health recommendations, on the other hand, do focus on added sugars, but I think you can see from those headlines that maybe we're kind of talking past each other. We're talking about added sugars as professionals, but the public is focused on all sugar, or they're not able to distinguish between the two. The dietary guidelines tells us to limit calories from added sugars. They tell us to consume less than 10% of our calories per day from added sugars. And the rationale for this is that if we don't do that, the chances of us being able to meet our nutrient needs while staying within the caloric recommendations or limitations that we might have to maintain our body weight are really, really difficult. So more from a weight management perspective. They also, if you dig deeper into the guidelines, they do mention that diets high in added sugars aren't consistent with dietary patterns associated with good health. So there's that broad health aspect as well. And we'll talk about what they call high intensity sweeteners. We've been calling non-nutritive sweeteners. You might know them better as artificial sweeteners, all kind of the same class of products here. But they actually suggest that if, hey, replace some of these added sugars with high intensity sweeteners, and you may see benefits in terms of caloric intake, and this could translate into longer term benefits in terms of weight management strategy. So public focused on sugar of all kinds, public health focused on added sugars, and we need to do a better job of educating on the important differences between those two things. Because if we don't do that, if we have people who are reading these headlines and thinking that fruits and vegetables are somehow bad for us, then the chances are that we're going to see an even higher number of people who are not meeting recommendations for these classic shortfall nutrients. So fruit typically has a taste or flavor profile of sweetness. Some vegetables have a flavor profile of sweetness, and some dairy products also have a sweet profile. And these are important sources of calcium, folate, potassium, magnesium, and vitamins A, C, D, and E. So it's important that we don't demonize sweetness to the point where people are making uninformed decisions about whole food groups and eliminating those. The other thing we need to remember is that sweetness isn't always an indicator that added sugars are present. It's okay, but there are a lot of foods that we eat that we might not realize could be contributing to added sugar intake. And for example, whole wheat bread. If I ask you your top 10 list of sweet foods, whole wheat bread is probably not on the list. But in some cases, and not all products, but in some cases, there is added sugar present. I found a particular product, the label said no added nonsense. But when I looked at the nutrition facts label, I saw that there were 3 grams of added sugars per slice. So I don't know what nonsense is, but apparently it is not sugar in this case. For example, if I have 2 pieces of toast for breakfast and a sandwich at lunch, I have already achieved about a quarter, 25% of the daily recommended intake for added sugars, and I haven't really experienced anything over the top sweet. So sweetness not always a great indicator. Same thing, another product I found, and I didn't have to look very hard, but this was an example of a chicken soup mix. Comes in a bag, you add the water to it and heat it up. 3 grams of added sugars per serving. Not something that you would really think, oh wow, this is too sweet for me, or oh, this has a sweet profile to it. And the last example, baked potato chips. Now the traditional old-fashioned chips probably don't have sugar added, but in this case the baked variety does. And some people might be reaching for this because they think it's healthier, and in some cases it is. But if we look at added sugars, this particular product had 2 grams per serving, and if you're like me, sticking to 1 serving can be a bit challenging. So before I know it, I'm increasing my added sugar exposure. So if sweetness doesn't necessarily reflect our exposure to added sugars, what's the point? Can we actually use information about sweet taste liking or preference that we might measure in the lab to predict dietary exposure to added sugar or anything like that? And the short answer is it's tricky. And this is because there are pronounced individual differences in sweetness responses. And that's where we're going to talk about sweet liking phenotype in just a little bit more detail than has already been mentioned. This slide is going to show you the utility, at least from a researcher perspective, of classifying people based on sweet liking phenotype. So I want to show you what this does and then tell you a little bit more about what it is, okay? So this graph on the left here shows you actual data that we collected from our lab. And on the y-axis along the bottom, x-axis along the bottom, we have sucrose concentration, and this is in percent. So we start off with a low percent, 3%, very mildly sweet, and we get up to, in this case, about 24%, which is fairly, it's sweet, no question about it. And what we're asking people is how much they like it, and we used a 15-point scale, and so they rated, the lower numbers, I like it less, higher numbers, I like it more. What you can see when we ask everybody is that there's this sort of increase from 3% to about 9%, that liking is increasing, and then it kind of levels off after that. So not a lot of change regardless of the concentration thereafter. Well, when we use some kind of fancy statistical techniques called hierarchical cluster analysis, not important for today's conversation, you get data that looks like this on the right. And while that might look like a giant mess, what it's showing you is that there are some very different response patterns to sweetness as we go upwards in concentration. And so we'll talk about this in this slide. Dr. Tepper has already explained to you that there are likers and dislikers, but I just wanted to hit on this idea of phenotype. That might be a word that you remember from intro biology or intro genetics, and it's been a while. A phenotype is just an observable trait, and in this case, we're talking about how much you like sweetness over increasing concentrations. And there are genetic and environmental factors that are at play here. We see in the graph here, we've got likers. Liking increases as concentration increases. We have dislikers, where as concentration increases, their liking falls off. They don't like sweeter things. And then we have a couple of other groups that show up from time to time, depending on the population that we're studying. We have people that we call the inverted U shape. They're in black here. And they like up to a certain concentration, but at that point it's too sweet and liking falls off as we get stronger in terms of concentration. And then there is a very interesting group of people, the neutrals, and they're in this kind of gold yellow color here. And you can see their liking doesn't really change based on concentration. They kind of like everything about the same. They're on a scale of 0 to 100, they're kind of at the 50 mark. And it's okay. I find these people really interesting because this is not the sensory world that I live in. I can definitely tell you if I like something that's more or less sweet, but these people really pique my interest, so worthy of further study. But anyway, these patterns show up time and time again. We have many studies where we look at this and find that there are these different responses to sweet liking. And so this is why when we lump everybody together in that slide where we just see kind of that slight rise and then the flat, it doesn't help us decipher or distinguish what's going on based on sweet liking. And what you might take from this, I mean, we're researchers. I'm interested in how taste is going to predict dietary intake. And what we find is that if we classify you based on your sweet liking phenotype, we are much more likely to be able to predict taste and diet intake relationships. So that's the utility of sweet liking phenotypes. So what are these differences? If I know a little bit about your sweet liking phenotype, what would I be able to guess about you? Well, it's probably just as you expected. Sweet likers are going to probably be exposed or consuming more added sugar. In the study that we did, we surveyed 418 people and found that sweet likers were consuming more energy from all beverages that we looked at, and more especially from sweetened juice and sweetened tea, and they were drinking less water than the other groups of people that we looked at, so the dislikers and the neutrals and so on. Another study found that sweet likers had increased preferences for sweet desserts. Sweet liking was positively associated with the intake of refined and total sugars, and sweet likers consumed more energy from beverages in another study. So this is a growing area of trying to associate taste and dietary intake, but what we find is that if we separate people out, we have a much better likelihood of seeing how their taste responses in the lab translate into behaviors in the real world. So what does this look like in the general population? How many people are sweet likers and dislikers and so on? We did a large study of about 1,000 people, 650 adults and over 300 children. And what we found is what you probably know if you are a parent, know a child, or have ever been a child. Adults and children have different sensory responses to sweetness. For the adults, we saw that about 1/3 of the population that we tested fall into the sweet liking category, and about 10%, a little less than 10%, were considered dislikers, and everybody else clustered in the middle. So they were either neutral or that inverted U shape. In kids, we don't see the neutral or the inverted U shape. We see a lot of sweet likers. About 80% of kids were found to be sweet likers, and about 20% were considered sweet dislikers. So kids and adults function in different sensory worlds, and this is, I think, a good indication of that. Now, how do I identify sweet liking phenotypes? Well, I bring you into the lab and I give you a bunch of concentrations of probably sugar and water, and I make you rate them, and then I put your data into the computer and I do some fancy statistics, and then all of a sudden now I finally know, oh, it turns out that you are a disliker. That's not going to work if you're trying to get this information from a patient or client in the real world, right, in your clinic. So some people have proposed a faster way to identify sweet liking phenotype. They use one solution, and they use a very easy descriptive way to do this. So let me just take you through it. They use a 34% sweet solution, sugar solution. It's 1 molar if you're, I don't know, keeping score at home, but 34%. Like Beverly said, this, you can feel the cavities forming in your teeth as you have this in your mouth. It's just crazy sweet. And what they do is they ask you to rate this on a scale. It's just a basic line scale with anchors of -50 to +50. And if you rate it as +15 or higher, you're considered a liker. -15 or lower, you're considered a disliker. And anywhere in between, you're one of those people that probably likes it up to a point and then falls off. So they surveyed over 100 people, and what they found is that in terms of that population, about 32% of their population were considered likers. Now, if you remember our study where we did the 5 different solutions and did the fancy statistics and takes forever, surprisingly close to what we saw. So about 34% in our case. And there is some difference between the U-shaped and the dislikers, but really we're interested more in identifying those likers so that we might be able to coach them about their exposure to added sugar and concentrate on maybe that as being a point of emphasis for education. I will say this brief method, the faster approach, does need to be more fully validated. We need to look at more diverse populations to see if this really holds up. But if so, it might be something that you could easily, you know, it's sugar and water. If you can make Kool-Aid, you can make up a test solution to do your own sweet liking phenotype testing. This graph just shows you very briefly the fact that while sweet liking phenotypes have been well established in study after study, most of those studies used sugar, especially sucrose and in some cases glucose, so nutritive sweeteners. There have been very few studies. One of ours is included here, looking at non-nutritive sweeteners to see if they also produce these sweet liking phenotypes, and in fact they do. So in our case, sucralose, what you can see here, the sweet likers in orange, and we collapsed all the other groups and called them non-likers, and they're in blue. And at the lower concentrations, the sweet likers like those solutions less than the non-likers, and at the higher concentrations, they like them more, so as we would expect. So just to recap, sweet liking phenotype, you know, it helps us to identify subgroups in the population. And if we know something about your sweet liking phenotype, then we can probably do a better job of predicting your exposure to added sugars and in the diet. Last concept, can't go anywhere without talking about non-nutritive sweeteners in terms of their ability to produce sweetness for us, but there's that disconnect between sweetness and energy. So if you remember, we think sweet taste is designed to identify energy in the environment. Well, here we have the signal, but no energy. And people have been concerned that that might trigger overeating because I'm getting the signal, but the payoff isn't there. So now I'm going to go out and eat some more stuff. And so what does the science actually say on this? So I'm going to just show you some epidemiological studies in a couple of different countries, since different countries have different food environments. We'll start with Australia. They used one 24-hour recall and surveyed over 12,000 people. And they found that free sugar, which they identify or describe as both natural and added sugar, was actually lower among people who consume non-nutritive sweeteners. Now, they didn't see any difference in energy intake, but in terms of free sugar exposure, that was lower. In the US, same idea, 24-hour recall, this time with over 22,000 people. And the authors reported that people who used non-nutritive sweeteners had a higher healthy eating index 2005 score. And this was because they were consuming less of the SOFAs, solid fats, added sugar, and alcohol. UK, there's two studies from the UK. They used 4-day diet records, so a little more in-depth information. One study looked at over 1,500 people, and the other study looked at over 5,000 people. And they were focused just on what the effects of non-nutritively sweetened beverage consumption, or things like diet drinks, what that did to diet intake and quality. And basically, similar to the other studies we've seen, people consuming non-nutritive sweeteners had higher dietary quality than people consuming sugar-sweetened beverages, or a combination of non-nutritive sweeteners and sugar-sweetened beverages. And the other study found lower energy and lower added sugar intake in people who were using the non-nutritive sweeteners. Now, in contrast, we see in Norway a different population and a different approach to measuring diet. They used a food frequency questionnaire, which is not as granular in terms of the specificity we can get with what's being consumed and how much. They also used a population of 100 people suffering from obesity class 2 or higher. And so these are very different people and a very different approach. But what they saw is kind of the opposite of what I've just told you. Non-nutritive sweeteners were associated with increased energy intake. Now, why this might be, is there something special about people with obese class 2 and above? Is it the tool that they used? Does that explain the differences? We don't know. And so this is an area that deserves further research. But right now, the preponderance of the evidence suggests that non-nutritive sweeteners may be a way to promote higher dietary quality. So to wrap up, we know that public health messaging now focuses on added sugar, which your patients and clients might be focusing just on sugar. So you have to educate them on the nuances between the two, because if they read those headlines and think that sugar is in fruit and sugar is in vegetables, then they might not be meeting nutrient requirements. I also told you that sweetness doesn't always predict poor dietary quality, and added sugars can show up in unexpected and really not so sweet places. There is individual variability when it comes to sweet liking, and we think that sweet likers are at more risk for consuming higher amounts of added sugar in the diet. And non-nutritive sweetener intake appears to be associated with higher diet quality in epidemiological studies, but this correlation does not mean causation, and so we really need to take a look at more of this data as it becomes available. So that's it for my presentation. Thank you so much for your attention, and I think we're moving to questions.